Hello viewers, welcome to my channel. Today we will discuss about the bacterial reproduction by binary fission. Bacteria, prokaryotic unicellular microorganism. They reproduce by budding, fragmentation and other means. Bacteria primarily reproduce by binary fission. It is a relatively simple type of cell division. The binary fission is an asexual process. In binary fission, a single cell divides into two cells. The steps of binary division is discussed here. Here, the diagram of the binary fission is also shown here. First, the cell wall, cell membrane and the overall volume of bacterial cell enlarges and the chromosome replicates. As the chromosome moves toward the opposite ends of the cell, the septum begins to grow inward. The diagram is shown here. The MreB protein may serve to move the chromosome to opposite poles. At this stage, other cytoplasmic components are distributed to the developing cells. The septum is synthesized completely through the cell center, and the cell membrane patches itself so that there are two separate chambers. At the last stage, the daughter cells are divided into two new cells. Some species separate completely as shown here, while others remain attached, forming chains, doublets or other cellular arrangement. As we discussed that septation is very necessary to divide the single cells into two cells. So here we discuss about the septation process. The septation is the process of forming a cross wall between two daughter cells. Septation is divided into several steps. First, selection of the site where septation will be formed. Then, assembly of a specialized structure called the Z ring, which divides the cells into two by constriction. Linkage of the Z ring to the plasma membrane, perhaps the component of the cell wall. Next, the assembly of the cell wall synthesizing machinery. Finally, the Z ring is constricted. Eventually, septum formation occurs. The assembly of Z ring is critical steps in septation, as it must be formed if subsequent steps to occur. The tubulin homolog FtsZ protein is found in most bacteria, many archaea, which forms the Z ring. The FtsZ protein polymerizes to form the filaments, which are thought to create a meshwork that constitute the Z ring. MinCD, together with other min proteins, oscillate pole to pole, thereby preventing the formation of off-center Z ring. Once the Z ring formation occurs, rest of the cell division machinery is constructed. First, one or more anchoring protein, such as FtsA, ZipA, link the Z ring to the cell membrane. Then the cell wall synthesizing machinery is assembled. The final steps in division involve the constriction of the Z ring, accompanied by invagination of the cell membrane and synthesis of the septal wall. The cell division apparatus is composed of numerous proteins that are thought to assemble. The process begins with the polymerization of FtsZ ring to form the Z ring. Then FtsA and ZipA proteins anchor the Z ring to the plasma membrane. In case of Bacillus subtilis, it is ZipA in place of ZipA. The function of cell division apparatus proteins in E.coli are shown here. FtsA, ZipA anchor the Z ring to the plasma membrane. The function of FtsEX is unknown. FtsK coordinates septation with chromosome segregation. FtsQ, FtsL, FtsB function is unknown. The function of FtsW, FtsL is peptidoglycan synthesis. The function of FtsN still unknown. The AMIC helps in the hydrolysis of peptidoglycan to separate daughter cells. Thus, in summary, in binary division, the cell elongates, chromosome replicates, and separate the newly formed DNA molecules so that there is one chromosome in each of the cells. Finally, septum or the cross wall is formed at the mid cells, dividing the parent cells into two progeny cells, each having its own chromosome and a complement of other cellular constituents. Thank you. Please like, share and subscribe to this channel.